Okay, so we're right on 1 o'clock, so let's kick off. Welcome everyone. So my name's Geraint Parry. I'm the Executive Officer of the Association of Applied Biologists, and I'm delighted to be hosting our webinar, which is hosted, as I say, by our Applied Tree and Forest Biology group. And we've got three really interesting talks on a diverse set of issues, all involving trees in some way or another. So I'm just going to give a little introduction before we begin about AAB. So AAB is a learned society that's based in the UK, but we organize activities internationally. And the applied biology in our context is really looking to improve agricultural productivity and sustainability. Our main activities that we organize conferences and different events, maybe up to 12 conferences each year. And then we own two publications as well. So we completely own Annals of Applied Biology, and you'll hear about Rob will be talking about a paper he published in Annals towards the end of the talk of the webinar today. And then we also third own Plant Biotechnology Journal with the Society for Experimental Biology and Wiley as well. So basically, we get some revenue from these journals, and then that all goes back into supporting the community through organization of meetings, providing travel grants and carer grants and different things. So the way that the AAB works is that we have 12, now actually 13 now specialist groups that kind of from the bottom up suggest activities with which the society should be involved, and then there's a small staff of us that are involved with that do all the organizing. So this meeting is organized by our applied tree and forest biology group, or at least it's motivated by that group. So we've just become involved with tree biology and forest biology over the last couple of years. We've organized 3 conferences in the last 4 years. One at Birmingham, one in Nottingham, and then one just a couple of months ago, which was in Cumbria, that have looked at kind of the research which will support like future policy and activities about management of individual trees and forests. And it's been a really successful arrival into the tree biology space, I think, for AAB, and this is something that we will be wanting to continue for years to come. So yes, so the 3 speakers we have are, as I said, across all different topics within this very broad brush of applied tree and forest biology. So just to advertise a few other things that AAB has coming up, as I said, we organize a lot of meetings, and if we've got a lot of tree people in the meeting today, then these might not be super relevant for everyone, but just to give an idea of the sort of things we do. So this month we have meetings on advanced integrated pest and pathogen management and regenerative agriculture, which are in Leicester next week, and then in York in a couple of weeks' time. So online registration, well, in-person and online registration still open for those. And especially we encourage early career professionals to register for these if you're interested. It's a very minimal cost to come to these. We have our annual Advances in Nematology meeting, which is in early December in London. And then we just launched pretty much today an interaction that AAB is now having with the Monogram Network. Some of you might have heard about that. So it's a network which looks at plants involved, small grain plants, monocots, and we're going to be organizing the meeting with them, which is going to be in their annual meeting, which is going to be in Cambridge next year. Okay, so go to the event website, the AAB events website to catch up with all these, and we hope to see you at an event coming up. So for the rules for today, obviously the chat box is available. So if you have any questions for our speakers, please write them into the chat box, or if you'd like to ask them in question, just put your virtual hand up, and we can bring you in to ask a question. We're going to record the talks, and they'll be shared on our YouTube channel after the event. And please look out for some information about our December webinar, which will be coming soon. This is going to be organized by our new early career professional specialist group. And I think we'll be looking at the do's and don'ts of giving short kind of elevator pitch style talks. So there's going to be a bit of tutorial and then some examples of that as well. So, Okay, so without further ado, I think we will move on. So our first speaker today is Estrella Luna Diez, who is at the University of Birmingham, and someone I like to think of as a friend and a colleague. I think, Estrella, you could say that. I think in these
Yeah,
type
we
of
are
things.
colleagues.
But so I've worked with Estrella on a project that she's going to talk about for the last few years. Sorry, working with her for the last few years, the project she's going to talk about now. So over to you, Estrella, to talk about Membra. Thanks very much.
Cool. I was trying to get myself out of the screen. Can you see me? Can you see my screen?
We can see it. You're not full screen yet, but
Yes, now I'm full
yeah,
screen,
you are.
right?
Yeah, yeah, yeah.
Okay, let's get rid of this thing. Perfect. Yeah, thank you, Geran, for the introduction. And yeah, we are definitely colleagues because we have been collaborating a lot on this project, which I'm going to present to you today. So I gave this title as Understanding Tree Memories for Climate Adaptation and Disease Resistance. But what I'm going to present to you today is all the different outcomes that we have had from a project that is called Membra. Okay, so maybe you have heard about Membra before. It's one of the Tree Escapes projects that it was funded in the first round, so it's a really large project. And Membra stands for Understanding Memory of UK Tree Escapes for Better Resilience and Adaptation. So we are looking at exactly how trees acquire, retain, and transmit memory of stress. So how we investigate this memory, or what we understand for this memory, are changes in epigenetics, specifically on DNA methylation. And maybe I've lost a big part of the audience by saying this, but I'm going to explain you exactly what this means. So epigenetics, it means things that are beyond the genetics, so above the genetics, okay? So we know that we have a DNA sequence, okay, with 4 bases, and they read information. They read phenotypes about us, about plants, about animals, about everything. But then there is something above all this DNA sequence, which is what we consider epigenetics, that are changes that happen to the DNA that basically open or close the chromatin, and the DNA is easier or more difficult to be transcribed. So one of these signatures of these changes that can happen is DNA methylation. So this is the addition of a methyl group to a cytosine. So when we have DNA methylation, then that sometimes happens at the promoter of the genes, and then that basically changes the activation of that gene because it's easier to be transcribed, and therefore that results in a phenotype. So a very key thing is that DNA methylation can be controlled by the environment. So whereas the environment cannot directly change the DNA sequence of an organism, the stress or environment stimuli can change the methylation profiles. And then something that is really cool is that whereas us in humans, we only have methylation when the cytosines are next to a G, okay? Plants have these really, really cool types of methylation that happen in different contexts, like for example, this one that we call CHG, so it means that there is another base in the middle that is not G, or even cytosines all on their own. So cytosines that are not surrounded by another G or anything. And this type of methylation is extremely, extremely dynamic, can be modified very easily by the environment. And this is something that plants have. So we are focusing a lot of our studies on this type of methylation. So now that you all know what methylation, DNA methylation and epigenetics means, and that is what we are going to be focusing on the memory, I'm going to talk to you about the stresses that we are looking in Membra. So we have different stresses that we classify as abiotic or biotic. So abiotic, we have drought, we have late frost, and also we have elevated CO2. And you may think, actually, elevated CO2 doesn't sound like a stress for a plant because it's basically fueling the plant growth. But we also know that too much of the good stuff can also be negative. okay? So that's why we consider it as stress, whereas it is more of a stimulus, but we consider it as stress. And then we have biotic stresses, like, for example, insect infestation events that basically destroy trees in a very specific moment, or ash dieback disease, which we all know that it has been devastating for the ashes in the UK. And then also we are looking at this complex syndrome that is called acute oak decline, or AOD, which happens in oak due to a combination of bacteria. So we have different hypotheses in Membra, and this slide can be a little bit daunting, but it is the essence of the project. So basically we have an overarching hypothesis, our hypothesis one, that is that stress results in a memory, okay, that trees can acquire memory. So we have changes in memory, that this is this DNA methylation that I explained you about, in the control, and the stress will have a different profile of DNA methylation. And then we have also other hypotheses that the amplitude of this change in the memory will depend on different things. So for example, we think that the more stress, or the higher the strength of the stress, or the higher the frequency, the more memory you will have. And this is something that it is relatively straightforward to hypothesize. But then we also thought that young trees will have a bigger capacity to acquire memory. So when you are a child or when you are young, then you can learn from your mistakes or the stresses that you had, whereas when you are old, maybe you don't care that much about remembering anything, because why would you do that? And also we have this hypothesis that is the life history strategy. So that means that trees that live very fast, or they are fast growers, then they will retain less memory than trees that take some time to grow. And we have this idea of live fast, die young kind of thing, that you don't remember that much about your past experiences. So in order to test this hypothesis, we have different experimental designs. We have drought that happened many decades ago, or it is happening right now. We have elevated CO2 that happened a while ago, or that is happening right now. Insect infestations that happen at a specific point of time. And then sadly, the diseases that we have because the trees do not recover are happening at this particular moment. We are doing the research in young and mature trees. In most of the diseases, like for example, not in acute oak decline, because it's a disease that only affects mature trees. And then we also have 5 tree species. We used to have 6 tree species, including a Sika spruce, but we decided not to spend that much money on the sequencing because it's a really, really large genome. So we have oak as the main species, but then we have ash, hazel, beech, and birch as well as the species. So I'm going to talk about a few things. We have all the samples collected in the project. The project officially ended in July 2025, so only a few months ago. We have all the individual species by condition prepared for publications, and I'm going to give you a little summary of some cool results that we have. So the first one, I'm going to start with the ash dieback disease results. So here we had young trees that were about 9 years old, which are very susceptible to ash dieback. And then all their trees, because I don't know if they can be classified as old, but they were about 50 years old. And what we did see, so we had 3 levels of disease. We had trees that were healthy, trees that had minor symptoms of the disease, and trees that were showing severe symptoms, and the same for the old. And what we can see is that actually disease results in a global methylation change. So especially in contexts like CG and CHG, we have an increase as the disease progresses in the methylation levels. That is not that obvious potentially in the CHH, although we do seem to see that slightly pushed higher with the severe categories. And something that is very interesting as well is that this change seems to be more pronounced in young trees, because here is where we see a very, very big change that happens, very similar to what we observe in all trees, like from healthy to the severe. We already have that big change there happening in the minor symptoms. But we also tested AOD, this acute oak decline, and this was in 200-year-old trees, about 200-year-old trees. So here the trees are a lot bigger. We had a very similar experimental design where we had trees that were healthy, trees that had minor symptoms, and trees that have severe symptoms. And again, the three contexts of DNA methylation. And here we cannot see any differences within the methylation percentages. So disease stress does not result in a global methylation change. These are different diseases completely. One is a fungus. This one is a multi-complex syndrome that is triggered by some bacteria, but other things can be impacting it. So we think that these diseases work differently, and also the fact that this is oak and the other one was ash. But in this system, we could not see any changes in global methylation. And then I'm also going to show you some results about the elevated CO2. And this is because in the Birmingham Institute of Forest Research, we have this fantastic facility that is called FACE. So that stands for Free Air CO2 Enrichment. And what we have is these constructions of tower cylinders that we call arrays. So each circle, each cylinder is an array, and we have 3 arrays that are just ambient CO2, and then another 3 arrays where we have elevated levels of CO2. In particular, we have the levels of CO2 that we will have in the planet in 2050. Here is mostly oak what we have, but we also have hazel trees growing and some other species, but our experiment here are focused mostly in oak. So we collected material from these trees that had been exposed to elevated CO2 for a few years, but we luckily also had some material from these trees from 2016, which is before the fumigation or the treatment of CO2 started. So what we have managed to do is that, because we collected material from the same trees, is that we have been able to identify changes in DNA methylation that occur in those particular trees that only were dependent on the CO2. So we compared the material that we collected in 2016 with the one that we collected in 2021. And that is something that has resulted in these regions that we call differentially methylated regions, or DMRs, that are pointing to specific parts of the genome where some methylation changes are observed. And tracking all those regions where the methylation occur, we can actually see that the most interesting changes are happening in CHH context that I told you was very dynamic, very exciting. And this occurs mostly in regions of the genome that are associated with transposable elements. So what are transposable elements? These are genes that are considered jumping genes because they move from one part of the genome to another. They are completely dependent on epigenetics, like, for example, DNA methylation. So this was a very exciting result to see that we are investigating further at the moment. Something along the way that it is not potentially that much dependent on the memory of the stress is something an observation that we got from our trees in the FACE facilities, but only looking at seasonal changes. So initially, what we did was to sequence the material from spring. So we collected material in late April, very early leaves. And then we saw that the methylation levels in CHH were really low, like were very, very low compared to what we used to see in other species or in other plant species. So then we decided to run as well samples in the same year, but from the summer and from the autumn. And actually what we see is a very, very clear increase in CHH methylation that occurs in summer and autumn. So this is a very good explanation, very good definition that the environment can shape DNA methylation at the CHH context. We didn't see that much this happening within the other contexts. We did see some things in the yellow bars, which are the progenies, but the biggest change happened at the CHH. So this is a really good example of how the environment can shape methylation levels. And something as well quite interesting is that actually seedling that is a few weeks old has a very similar profile of methylation at CHH than a 200-year-old tree in summer or in autumn, which is something that for me is mind-blowing. So the result summary so far that we have is that in terms of hypothesis one, okay, that elevated CO2 does not affect global changes in DNA methylation, but there are changes specific in different regions which are highly interesting, are these jumping genes. Also that as-dieback disease results in global changes in methylation. So we do have some stresses that are impacting the methylation. The hypothesis two, it was that increased methylation is dependent on the strength of the stress, and that's something that we see with ash trees, specifically when going from the healthy to the severe. And then also we have that hypothesis four, that older trees show lower levels of basal methylation and young ash trees show significant changes. So this is potentially something that is going to reflect a higher memory imprinting of those young trees. And finally, that changes due to disease severity seem to be different between ash and oak. So we have change in DNA methylation or in the memory with ash, but no with oak. So that's potentially something that reflects as well the life history strategy between ash and oak. So what we are doing at the moment is we are looking at the progenies because we collected material from all these trees. We want to know whether actually these changes in methylation are actually passed to the progenies and whether the progenies have different phenotypes to those stresses. So we have a few papers in preparation at the moment, specifically with the elevated CO2 and the drought. That is where we see very important changes in the progeny phenotypes. We are looking at the identification of these differentially methylated regions. And finally, what we are doing now it is to link the results with forest dynamics and ecological models. And this is what I'm going to focus the last part of my presentation. So this project has been an integration of different disciplines, okay, within the, I have a question. Within the ecology and the biology, and also the arts and humanities, but especially the ecology and the biology, we have been very interconnected. So why we decided on those stresses was not random. It was because we know that those stresses affect forest dynamics, okay? They've changed traits in trees. So for example, drought affects growth, the same that insect infestation or disease with as-dieback affect growth, okay? So now we know, if I can go through the next slide, what's happening? Okay, there we go. Now we know that epigenetic legacy of trees exposed to these events or these stresses change that epigenetic legacy. So we know what the epigenetic legacy is. So we are basically integrating long-term monitoring of data on forest dynamics with this DNA methylation data. So we have different experiments where we have been tracking, well, not me particularly, but other people have been tracking for decades how those trees have been growing. So that now is what we want to integrate with this DNA methylation data. And we have different routes of this integration. The first one was simply to look at all these regions, you know, these DMR, differentially methylated regions across all conditions, okay, and identify overlapping regions within the different species. So we started doing this first with oak. So we have the differentially methylated regions or the effect in methylation that occurs from drought, from elevated CO2, from insect infestation, and from acute oak decline. And then we identified that there were 12 regions in the genome that actually show a commonality. So we have been identifying the levels of methylation that we have in those regions, and we have been able to see that stress trees seem to have lower methylation of the control. So that is what the change in methylation is a decrease in methylation. And in particular, there are 3 regions in the genome that are hypomethylated when stressed, and these are these 3 regions that we have here. Okay, so what we are doing with this information now is that we did a new collection campaign within the plots that we have been monitoring long term, and we collected 60 oak trees that were either winners or losers. Some of them were stable. So we consider a winner a tree that is been growing really well, and a loser that for some reason that tree hasn't been growing that well. So then we did an analysis of targeted methylation in those trees, and actually we don't see a major change, but we do see some trends that indicate that the biggest winners, so the trees that were even the top winners, are associated with unmethylated sites. So this is a little bit of a contrasting or conflicting result, because we did see that the trees that were stressed were unmethylated, and now we have the winners being unmethylated. So the idea would be to collect more trees and to do a much larger analysis in order to identify whether this is a trend or it is something that it is real. How we are getting out of oak, it is a very complicated thing, but we are attempting to do this with synteny. So basically, synteny analyzes regions of the genome or identify regions of the genome in oak, in other tree species. So we have here the similarities that we have with birch, hazel, and beech, and ash is the farthest away from oak. So the idea would be to, once we have identified those regions fully, then we can look for those regions in other species. And then we have another route, which is a lot more complicated in a way. However, I'm going to present it to you just to give you an idea of what we are doing at the moment in that integration. So basically, we have the genomes of 5 tree species, okay, which are relatively different, but especially ash, but there is a commonality of genes that are homologous to all those 5 tree species. So we basically have put together all these genes, it's about 9,000 genes, and of these 5 tree species, and then we have looked at the methylation patterns on those genes. And what we have been able to identify is things like this. I'm presenting the one at the CHH context, as I told you, but we have exactly the same with CAG and with CAG methylation. So we are able to identify some patterns of changes of DNA methylation that are shared. So this is just the matrix that we have created. There are some very obvious things, like for example, the oak and beech hypomethylation, especially here where the spring samples are. Some beech specific hypomethylation going on here as well. But this is just basically, we are scratching the surface of what we can do with this data set at the minute, because we have the methylation of all those species plus all the homologous genes. So now it's all a matter of looking much more deeply into this data set. So these are the 2 routes that we are taking. One is like much more straightforward, but potentially with a lot more artifacts and noise. And this one is the one that potentially can give us a very good answer, although we will be missing some specific things that will happen to each tree species. But here we will be able to identify regions that are shared across a tree species and that also change in methylation due to stress. And with this slide, I'm finishing the presentation. Just to thank everyone involved, specifically my lab. So this is a picture of our 2025 Lab Away Day to Kew Gardens. Rosa is the research postdoc that has been very involved in the project, the same than Marcella and Alicia, who is an MRes student working in the project, and Joe, that was the bioinformatician. I also have to thank, of course, all the By4 Tech team and former students that have contributed so much into this project. And then you may be wondering what on earth is going on here. So this is my colleague, Marco Catoni, who is the person that is an expert in epigenetics and DNA methylation, especially at a computational level. And we are doing yoga. We are doing yoga in the forest. And the reason for this is because, as I said, Membra is a really multidisciplinary project that we are also involving the arts and humanities. So one of the aims of Membra is to try to understand how the perception of humans, of trees, change when we know that trees have memories. So do we value them more? Do we see them differently when we know that they actually remember that stress? So this is part of one of the initiatives that we've done together with the artists. Of course, I have to thank the funding institutions, the Tree Escapes program, and that's it. I thank you for your attention. If you have any questions, I'm happy to take them.
Brilliant. Thanks very much, Estrella. That's great to hear a summary from Membra. And I think it's difficult to underestimate that that people might have worked on methylation or in other plant species. They don't know anything until they come to work with trees and the extra complexities there are in that. So we have a couple of questions already come in. I can ask those for you. So from John, who's going to be speaking next. So he says, Estrella, are you able to see any gene families particularly represented in the differentially methylated regions versus in unaffected versus severely affected trees? So I think there's quite a short answer to this, but Estrella, over to you.
Yeah, the answer is yes, of course. Yeah, we can see definitely some specific families. Whether they come with immune responses, it happens specifically in the seedlings, like in the progenies, actually, is where we see all the things coming with the immune responses and the differences in the DMRs. We do gene ontology analysis. So as soon as we have all the DMR regions, when they target genetic regions, genetic features, then we automatically do gene ontology, and then is where we are able to identify all these families. Yeah. Mm-hmm.
So we have a question from Romain, a bit more of a technical question, I guess. So does the tissue selection for sampling affect the levels of methylation? So samples from asymptomatic or symptomatic leaves from the same individual?
Yes. Yeah, I mean, this is a fantastic question, and this is one of the things that we have been trying to address as well, because we have always been sampling from leaves, and we try to sample always from the same position in the canopies. So we go mid-canopy. The arborists do this work. They are fantastic to keep things reproducible. Now, whether they are affected or not, then this is a bit complicated because when it comes to us dieback, usually if they are affected, we don't have so many leaves. So we usually tend to have asymptomatic leaves, leaves that are healthy. When it comes to, for example, oak trees, we do have quite a few things going on in the oak tree leaves. We have powdery mildew over the summer and in the autumn. We also have some insects. So these are trees that they are exposed to everything. So we always try to get the same type of leaf that are unaffected by things. So we try to keep the experimental design as clean as possible, but this is one of the most important points. At the present time, something that Rosa is doing is that together with Marco is that they are optimizing sampling of the tree trunks from the wood because they want to be able to extract DNA and do analysis of methylation from the tree rings. So we could actually track how things change when the stresses happen. So, but that's something that they are still optimizing.
Okay, so another question from Dario. who says, very cool talk. I know this is probably an unsolvable problem, but your ash dieback induced methylation changes have a very small effect on effect sizes and are more clear in the CG. How do you know that these are induced by disease rather than having sampling from trees that have lower levels of or different levels of starting methylation rate?
This is a fantastic question, Dario. This is something that we have been encountering as a very problematic thing. Like, is this something that the tree, for example, has different levels of methylation just because of tree diversity? Or is it because maybe the trees were less affected by the disease and not as a sign that they were more resistant? So what we have done now to actually clean the noise from the ash project is that we have taken material. So we took material in 2021 from healthy trees, minorly affected and severely affected. And then now, last year in 2024, what we did is we went back to those trees and we collected material from the trees that were once healthy. Now they are showing minor symptoms, and the ones that had minor symptoms, they are showing severe symptoms. So now what we are doing is to control for that genetic difference and isolating the effect of the disease. And this is something that we are currently analyzing at the moment. We can see these increases in methylation as the disease progresses as well, but the specific regions and things is something that we are still identifying, but this is a bang on question. Yeah, very good.
Great. Yeah, thanks for the question, Dario. So let me ask a final question, Estrella. So maybe the clearest result you showed was about the changes in methylation within transposable elements.
Yeah.
So in response to elevated CO2. So from your knowledge of other plant systems, can you suggest some sort of mechanism that might be happening here in the trees?
Yes, I mean, we do have definitely a lot of research done with transposable elements in tomato, for instance, and everything to do with induced resistance and how can different phenotypes? I mean, we do know that age and environment and growth during the life of a plant or the season, it is majorly regulated by transposable element expression. And we do know that there are quite a lot of transposable elements in oak. However, this is all a very big speculation, but it could potentially be that those changes in DNA methylation are occurring in order to silence those transposable elements to a way of controlling, growth as it happens throughout the season. And that is because elevated CO2 is affecting directly growth. So we see more growth under elevated CO2. So that is potentially one of the mechanisms that plants are being affected in when they grow more after that exposure to elevated CO2. But this is a really big speculation. That would be a hypothesis, basically.
Yes, and as you know, having those sort of hypotheses in tomato or Rhabdopsis, you just grow another generation, and you can do it less easy when you're looking at 50-year-old oak trees. That's for sure.
Yeah.
Okay, brilliant. Thanks very much,
Thank
Estrella.
you so
So
much.
there's one more question in the chat. If you could
Yeah.
if you could answer that in the chat, that would be.
No, we didn't take transcriptomic data actually. Yeah, that is a really good point. So we do have some transcriptomic data that it's potentially going to happen in the next few years, but we didn't look at correlating the methylation with the expression. But that would be a really important point, but it's a bit out of the scope of the project. I'm sure a lot more money in sequencing.
Okay, great. Thank you very much, Estrella. That's great.